Okay, good morning, everyone. Happy Monday. So a couple of announcements quickly before we get started. Labs and discussions begin today. Please attend your assigned section, and if you need to change, there's instructions in the handout that was given on the first day and on B-Space for how to do that. And my office hours, I'm going to be changing the location, so watch announcements on B-Space. I'll probably be posting that by later today, and an email will be sent to all of you for the new location for my office hours, so we have a little more space. Okay, so today we're going to start talking about cell structure. We have 2 lectures on this. It's obviously a very big topic. It's a big field in biology, and there's a lot of exciting research going on in this area, and I'll try to highlight a few of the things that have happened recently in the field that are, I think, incredibly interesting and exciting as we go along. So when you go to do the reading in the book, please pay particular attention to the pages that I highlight in the handout. I'll always tell you on the first page of the notes for lecture which pages in the book I would like you to refer to that will directly pertain to the material in that lecture. So that's the most accurate place to go for the reading that you need to do. Okay, so today we're going to talk about methods to study cells, and then we're going to talk about the differences between the 2 major types of cells in biology, prokaryotic cells and eukaryotic cells. And then we're going to talk about protein trafficking and organelles. Okay, so it's a lot, and we'll try to do our best to be clear. So first of all organisms are made of cells. You probably know this. The cell is the simplest collection of matter that can be alive. Okay, so is a virus alive? Is a virus a cell, first of all? Does anybody know? Is a virus a cell? No. Is it alive? No, right? Because it's genetic material that really has to be, it's dependent on a cell for replication, right? A virus can only propagate more viruses by infecting a cell. Okay, so viruses are not really considered to be alive. Cell structure is correlated to function, and this is why we pay a lot of attention to the actual structure inside the cell. And very importantly, all cells are related to their ancestral cells, right? So all cells are descendants of other cells. They're copies of other cells genetically. Okay, so that's a very important underlying theme in everything we'll be talking about in this course, is the evolutionary relationships. Okay, and so before we can really talk about what's inside of cells, we really have to think a little bit about how we study cells. And this is an area that's been incredibly interesting and exciting in biology. Lots of really cool technological developments, certainly since the time that I took this equivalent course in college in the last 20-odd years. A lot of really great work has been done to improve the quality of the kinds of techniques that we have to study cells. And you'll see a lot of that in your book highlighted. And so the 2 methods that we're going to highlight here are microscopy and biochemistry. So these are 2 tools that have given us a lot of information about the way that cells look and, most importantly, how they work, how they function. Okay, so let's talk a little bit about microscopy. So scientists use microscopes to visualize cells that are too small to see with the naked eye, right? So in most cases, we can't observe cells with our eye. We have to have help to do it. And the simplest type of microscope is called a light microscope. And you'll see this abbreviated in your book, LM. So if you're going through the book and looking at some of the gorgeous pictures in there, they will always abbreviate the micrographs that are taken from a light microscope with LM. So that's what that is. And the way these work is that visible light passes through the specimen, which is typically on a glass slide, and then through a glass lens, and the lens will magnify the image so that we can observe it. And the quality of the image depends on a few things. One is magnification, the ratio of the size of the object to its real size. And secondly, resolution. So resolution is a measure of how far apart we can resolve 2 separate images or 2 separate objects in an image. And the third aspect is contrast. So the better the contrast, the better we can observe individual features in the cell. And a lot of the advances that have happened in microscopy in the last decade or two have really been advances in this third thing, contrast, really improving the way that we can provide contrast in samples. Okay, this is a nice chart from the book that just kind of shows you at a glance the levels of sort of sizes of things that we might like to look at, what we can see with the unaided eye, what we can observe using a light microscope, and then what we would have to use another type of microscopy for, a type of microscopy that's called electron microscopy. Okay? And so with a light microscope, you can observe samples that are down to about 0.2 microns in size, roughly 0.2 microns. So that means that if you look at this chart, we can observe plant and animal cells pretty easily using a light microscope with scopes that are a little bit better, higher resolution, good staining, good contrast. We might be able to observe things inside the cell like the nucleus, bacterial cells. And then this is kind of right at the edge of what we could see by light microscopy would be the observation of organelles in the cell. These are, we'll talk about these today, but these are smaller units within the cell that have very important functions like the mitochondria, and those would be a lot harder to see by light microscopy. And then down here, for the smallest bacterial cells, we actually would need to use an electron microscope to visualize those. Viruses are smaller than cells. As we discussed, they infect cells, so they're smaller. So we would need also to use an electron microscope to see viruses. And then other things in cells like ribosomes, proteins, lipids, and then smaller molecules eventually get down to the range where even the electron microscope isn't good enough to observe those, right? But you can see that with using different types of microscopy, we can actually get a large range of magnification so that we can actually observe most of the kinds of things in the cell that we'd like to see, the structure and even some of the individual molecules if they're big enough. Okay, so light microscopy, so these kinds of microscopes can magnify effectively up to about 1,000 times the size of the actual sample. Okay, so that's useful to know, sort of as a rough reference. And as we mentioned, various methods are used to enhance the contrast so that we can see the specimen. And so if we're looking at a cell, I don't know if you, how many people have looked at a cell under a light microscope? A lot of you, right? Yeah. So, you know, I took my son to the Denver Museum of Natural History last summer, and they had a great program going for kids, and kids like me, too. I thought it was really fun, where you could, they had a bunch of light microscopes set up, and you could go in and do an experiment. And the experiment was to visualize your own cells and figure out look at the nuclei and see if you could see any other structures in the cells. And these were just very simple microscopes. And the experiment was to take a toothpick and just scrape a few cheek cells off your cheek and then, you know, smear them on the slide and look under the scope. And so we did this, and my son looked under the scope and he said, "I don't see anything. You know, it's blank." And I looked, and you know, you could see that there was something there, but you really couldn't visualize it. And then he realized, oh, I have to follow the protocol. And the protocol was to take a little eyedropper full of stain and stain the sample. So that's called fixing the sample. And once the sample was stained, and then you put it under a little heat lamp to dry it off, put it back under the scope, voila, beautiful, you know, gorgeous cells. You could see the beautiful cell membrane on the outside and very dark central spots that were the nuclei, because they stained very darkly with this particular stain. So that's something to keep in mind, that with the light microscope, sample has to be fixed, which means that the cells are not alive anymore, right? So we're looking at a dead cell, and we have to apply a stain, which means that the stain might have an effect on the sample, right? It could affect what we're seeing in the microscope as well. So just a couple of caveats to keep in mind. This is true actually for any type of microscopy where we have to add some type of stain to the samples. In some types of microscopy, we don't have to do that, but when you have to, then it introduces a variable to take into account. And then importantly here, and I mentioned this before, but most subcellular structures, and these would be things like the organelles that we're going to talk about, mitochondria, chloroplasts, vacuoles, other sorts of internal structures in the cell, are often too small to visualize very well by light microscopy. You can see they're there, but you can't see any detail. And this is just an example. So this is basically sort of what I told you before, that if we use the simplest form of light microscopy, which is called bright field microscopy, if we look at an unstained sample, you can see it, but it's not very easy to make out any of the internal structure of the cell. Once we apply a stain, now we can see very clearly the membrane of the cell. You can sort of see the folds of the membrane. We can see the nucleus in the center very clearly, and then you can see that there's a lot of other stuff in there. You just can't really resolve what it is. Okay, and so people that were developing microscopes, so the light microscope was first developed in 1590. Amazing, right? A long time ago. And in the 1600s, the techniques were really improved quite substantially. And then it wasn't until the 1950s that electron microscopy was developed and started to be advanced. And one of the motivations for developing an electron microscope was the realization that the limitation in light microscopy is really the wavelength of visible light, right? So we're dependent, we're sort of limited by the resolution that we can get by the wavelength of visible light that can pass through the specimen and be focused by the lens of a light microscope. For an electron microscope, we have a much smaller particle, much smaller wavelength, and so we have the potential to image samples at a much higher resolution. And so that was a very strong motivation to develop this technology. And so these types of microscopes are used to study subcellular structures. There's a number of different types of electron microscopy. If you go to Wikipedia and look up microscopy, they have actually a very nice article there, I thought, about microscopy, and they talk about a number of different types of microscopy. But for the purposes of this course and for thinking about the kinds of pictures that you'll be seeing in the book, I really just want you to be aware of two types. One is scanning electron microscopy, which is abbreviated SEM in your book. And this is a type that focuses a beam of electrons on the surface of a sample and provides images that look three-dimensional. And so the way that these actually work is there's a little lever arm in this scope that is basically tapping across the surface and interacting with the surface of your sample and mapping out a surface structure. And some of these images are just, I think, really breathtakingly beautiful. There's some nice ones in your book, but there's even nicer ones floating around on the web if you go looking. And then the other type of microscopy is transmission electron microscopy. And these are microscopes that focus a beam of electrons through a specimen. And so these are what we use typically to study the internal structure of cells and really look inside of cells. And so a lot of the images that you'll see in the book are using transmission electron microscopy. And these are just a couple of pictures. This is actually right out of your book. This is a scanning electron microscope image of the cilia on the surface of an animal trachea. And this is showing these little hairs that are basically waving on the surface of these cells and are responsible for moving debris and particles through the trachea. And you can see that in a scanning picture like this, we see the surface of these waving fibrils or cilia, but we don't see inside the structure of these guys. If we look down here with a transmission electron microscope, now what's done is to slice through the sample, and we can actually see inside. And you can see that when you make a slice like this, some of the cilia are sliced in the long direction, longitudinally, so we see what they look like in this direction. Some of them are sliced horizontally, so we see what they look like in cross-section that way. So it's kind of nice. You see both images. And then this is not in the notes, but this is taken off of this Wikipedia page, gorgeous picture of pollen grains taken with a scanning electron microscope. And look at that. I mean, it's just really beautiful, the kind of resolution that you can get. So we get very accurate images of what the surfaces of these different types of pollen grains actually look like. Couple things, yeah, I think I already mentioned this. This is typically you can get 200 to 250 times better resolution than you could see with a light microscope. The sample for these types of imaging methods these samples have to be dried and they have to be fixed. So just again to keep that in mind, that could introduce changes in the structure that you're studying. Okay, and the other method that I want to mention is cell fractionation. So this is a method in which cells are taken apart, and then we can use centrifugation to fractionate the contents of the cells and study these individual fractions. And if we put this type of biochemistry together with the kind of cell biology or cytology, really, that we've been talking about, where we're just looking at what's inside cells, we can really get a very accurate idea of what's happening in the different parts of the cell, what molecules are there, what their functions are. And that's really sort of the combination of these two approaches that makes up the field of cell biology. This is a slide, again, this is from your book, that just shows the process of fractionation of cells, where we can take a sample of tissue, or these could be a bacterial cell culture, for example, or it could be some leaves, or mushrooms, or it could be anything, right? And we could grind it up and then fractionate the sample by centrifugation, where the heaviest particles will spin to the bottom of the tube, and the lighter particles will end up higher up in the tube. So we can fractionate things that way and isolate those fractions. And then depending on how far we want to go with it, we could even get to the point of purifying individual molecules out of that sample and studying those molecules. And so a lot of what the research in my own laboratory, for example, involves that level of research, where we're taking cells, we're breaking them open, we're purifying individual kinds of molecules, and then studying how they work and what they look like. Okay, so now we want to, now that we sort of understand what the methods are that have been used to do a lot of the research and make the kinds of discoveries that we're going to discuss, I want to move on to talking about the different types of cells. And really there are two kinds of cells in biology: prokaryotic cells and eukaryotic cells. Okay, and so first of all cells share some common features. So all cells have a plasma membrane outside the cell. All cells have inside the plasma membrane a substance called the cytosol, right, or the cytoplasm. All cells have chromosomes that carry the genetic information, the genes. And all cells have ribosomes, and the ribosomes are the factories that make proteins from those genes. Okay, so those are four features that would characterize any kind of cell. But then it turns out that if we look in a little more detail, there are two types of cells that are called prokaryotic and eukaryotic, and these have some very interesting and important differences in the way they're set up and the way they work. And we want to talk a little bit about that. So you probably know that there are three kingdoms of life, right? There are bacteria, archaea, and eukarya, right? Three kingdoms of life. Two of those kingdoms, bacteria and archaea, consist of organisms with prokaryotic cells. Okay? The eukarya are organisms that have eukaryotic cells. And this just shows you that protists, fungi, animals, and plants, these all are eukaryotic cell organisms and have eukaryotic cells. Okay, and so the next two slides just tell you a little bit of the features of these different kinds of cells. So prokaryotic cells don't have a nucleus. This is one of the most obvious ways that we can tell, just by looking at a cell, whether it's a prokaryotic cell or a eukaryotic cell. So bacterial cells, for example, they don't have nuclei. Instead, the DNA is in an unbound region, so it's in a region of the cell that isn't separated from the rest of the cell by a separate membrane. And it's called the nucleoid. So it's a region where there's a concentration of DNA. And there's a lot of work in the microbiology community has shown that, in fact, it used to be thought that the DNA was just kind of, randomly sitting there like spaghetti. But in fact, it turns out that the bacterial DNA is actually packaged by proteins that resemble the histone proteins that package eukaryotic DNA. We'll talk about that more later, but just to keep in mind that the DNA is it's organized in those cells. There are no membrane-bound organelles in bacterial cells, and the cytoplasm is bound by the plasma membrane. Okay, and so this just shows a sort of an artist's rendition of a bacterial cell. These are typically rod-shaped cells. You can see there's little cilia, they call them fimbriae, on the outside of the cell, and then there's these flagella that help the cell to move. Here's the nucleoid where the DNA is concentrated, and then over here is an actual transmission electron micrograph of a bacterial cell with these regions of the inside the cell indicated. Here's some ribosomes, for example. Okay, and then so here's, in contrast, is a picture of a eukaryotic cell. So eukaryotic cells have DNA that is contained within a nucleus, and the nucleus has its own membrane. It's called the nuclear envelope that encloses the genetic material. And the DNA inside the nucleus is in the form of chromatin. So the DNA is, again, it's not just stuffed in there, it's actually packaged very carefully by wrapping around proteins that are called histones. And so the DNA is kept in a very organized way there, and there's a lot of research has gone into understanding how individual sections of the DNA that contain genes that need to be transcribed into RNA and then made into proteins, how those regions of the chromatin become exposed at the right time so that they can be read out by the machinery in the nucleus that is responsible for making a messenger RNA and then getting it out of the nucleus. Eukaryotic cells have membrane-bound organelles. We'll talk a little bit more about that later in the lecture today. And then the cytoplasm is the region between the plasma membrane and the nucleus. And so if you just read this and you didn't look at the picture there, you might get the idea that the cell is kind of empty, right? It's sort of, it's got this big space in the cytoplasm, and it's got a nucleus somewhere in the middle of the cell, and maybe there's a few organelles floating around. But if you look at this, again, an artist's drawing of the cell, you get the sense that, in fact, these cells are incredibly dense, right? They're very densely packed. There's a lot of stuff in there, and we're going to talk about what some of that stuff is today. But a couple things to point out. So here's the nucleus here, bounded by the nuclear envelope. Inside the nucleus is a concentrated region called the nucleolus, and this is where ribosomal RNA is made and processed. And it's also the site where there's a lot of organization of other RNA protein complexes that have to be assembled in the nucleus. That all goes on in this region called the nucleolus. And then you can see that the membrane is surrounded by this blue sort of variegated region here, which is called the endoplasmic reticulum. And we'll talk more about that in a moment. And then outside of just beyond the endoplasmic reticulum is the Golgi right here in this sort of lighter green color. And then over here we see some organelles. Here's the mitochondrion sort of cut in half, and here's one over here. Here's another one. And then here's a centrosome. This is the region where the chromosomal DNA comes together during cell division and the way that the chromosomes are organized. So the cell divides and puts the right pieces of DNA in the right daughter cells as the cell is dividing. And then there's some other things here that we'll talk about, the lysosomes, peroxisomes, and then on the outside of the cell, these little microvilli that can be protruding from the cell surface and are thought to be important for increasing the surface area of the cell, especially for a secretory cell. That could be very important to help the cell have more contact with its surroundings. Okay, and so something very important to notice here, eukaryotic cells are generally a lot bigger than prokaryotic cells. Okay, generally a lot bigger. And so in doing microscopy, it turns out that initially it was a lot easier to image human cells, scraped off of our skin or our cheek, than to try to look at bacteria, because the bacterial cells are just a lot smaller. Okay, so let's talk now a little bit about what all these things are inside the cell, inside the eukaryotic cell. So as we said, the nucleus contains the cell's genetic material. It's usually the most obvious organelle in the cell, and you saw that picture just from the bright field microscopy. You could see the nucleus very clearly. And one of the reasons for that is that when you do staining of these cells, the stains will often interact either with the nuclear envelope or with the DNA specifically, and allow us to visualize the nucleus a lot more easily than other parts of the cell. The nuclear envelope is what encloses the nucleus. It separates the nucleus from the surrounding cytoplasm, and this is a double membrane, right? So it's a double membrane, and each membrane consists of a lipid bilayer. Remember, we talked when we talked about lipids, we talked about phospholipids, and those phospholipid head groups with their little fatty acid tails hanging down, forming a double layer, right, a lipid bilayer, and there's two of those going around the nucleus. And then there are pores that are responsible for controlling the molecules that get in and out of the nucleus. And these are called nuclear pore complexes. And they're very big at a molecular level, and they're very complex. They have many proteins that comprise those nuclear pores. And again, lots of work has gone into trying to understand how they work. Very interesting. Ribosomes, which are some of the biggest macromolecules in the cell, have to go across the nuclear membrane. They have to get in and out of the nucleus, or at least out, and so they go out the nuclear pore. So these pores have to be big on a molecular level. And then finally, the shape of the nucleus. So the nucleus isn't, again, it's not just a wiggly bag. It actually seems to have a real shape to it, and that's because there is something called the nuclear lamina, composed of protein that provides some internal structure to the nucleus so that it doesn't just collapse in on itself. Okay, and then here's some pictures of some of these things. So this is showing us a picture of the surface of the nuclear envelope. This is this is a really nice image. It's actually using a method called freeze fracture for microscopy, in which you take the sample and you quickly freeze it, so that you freeze it in such a way that ice crystals don't form, so it doesn't damage the sample through ice formation. And then the sample can be fractured in the sense that we can, if there's a lipid bilayer, like there is, in fact, two lipid bilayers going around the nucleus, we can actually peel one of them back. And you can see there's kind of a line right here, right? And so this is where this fracturing has occurred. And so we can see what's inside and outside of this membrane. And so here's the nuclear envelope. You can see this is the outer membrane. Over here is the inner membrane. Here's the fracturing line separating them. And then you can see little indentations on the surface that correspond to the nuclear pores. And here's the artist's rendition of this. So you can see that the surface of the nucleus is just studded with these pores. They're everywhere, right? There's lots of these things. And that's because there's lots of molecules, there's lots of molecular traffic that has to be able to get in and out of the nucleus. And that is all carried out by these pores. This is, again, a sort of a drawing of the close-up of the nuclear envelope, and then these pores with ribosomes approaching and getting ready to go across. And here's an actual transmission electron micrograph of some of these pores. Now, I was just at a structural biology meeting a couple of weeks ago where there was a lot of discussion of the structures of nuclear pores. And one of the fascinating things that came out of this meeting is that even though nuclear pores, it would seem like they have to be incredibly they've been around a long time in evolution, right? Ever since, cells had nuclei with molecules that had to get in and out, there sort of had to be nuclear pores to do that. So you might think that, if you were just guessing, that nuclear pores would all be very closely related to each other in different kinds of organisms and different types of cells and different types of nuclei. But it turns out that, in fact, the structures of nuclear pores are really different in different types of cells. And nobody really knows why. It may be that there's a lot of evolutionary pressure for them to evolve quickly so that they can specialize for controlling the entry and exit of different kinds of molecules. So just something that I thought was surprising to me. It was really, really interesting. And these structures are incredibly beautiful if you look at some of the images that have been developed for them. Okay, ribosomes. So again, all cells have ribosomes. These are the machines that make proteins in all cells, and they're made up of protein and RNA. So interestingly, in ribosomes, about 2/3 of the ribosome is RNA, and the other 1/3 is protein. And the ribosome is actually it's a particle that has 2 subunits to it. The 2 subunits have to come together to make proteins. And the location of protein synthesis in the cell is in the cytoplasm, but it happens in 2 different locations in the cytoplasm. One is in the cytosol, where the ribosomes are actually free in the cytosol, so they're not attached to any membrane surface. There probably is there may be some organization to them, but they're not sitting directly on a membrane. And the other location is on the outside of the endoplasmic reticulum, or the nuclear envelope. And these are ribosomes that are actually attached to those surfaces, and protein synthesis is going on those surfaces. And we'll see some images of them, what they look like there. Right here. Okay, so here's a transmission electron micrograph showing the endoplasmic reticulum, these lines going across here. You can see this membrane. And then we see in between a whole lot of free ribosomes, and then on the surface of the endoplasmic reticulum we see a number of bound ribosomes. And you can see that, in an image like that, we're not seeing a close-up at all of what those ribosomes actually look like. We can see they're really dense. Anybody know, like, how would I know how do we know those are ribosomes and not something else? It's kind of a hard question, actually. So, I mentioned that the ribosome is 2/3 nucleic acid, right? So nucleic acid tends to be a lot more electron dense than protein. It's got a lot of phosphates in the backbone, for example. And so there are stains that one can use that will preferentially stain nucleic acid. So that's one of the ways that we can do it. I forgot to mention this earlier, but a lot of the electron microscopy that you see, again, we have to use stains for those, right? And typically there we're using metal-based stains that will be taken up by certain components of the cell, and then electrons will interact preferentially with those metal-based stains. And so that's another way that we can do it, is to use stains that preferentially will interact with nucleic acid. And we know that the most, the particles in the cell that contain the most nucleic acid are actually ribosomes. And then your book had, I replaced this. So it's very sad to me that, your book had this kind of little blobby drawing of a ribosome there, and Mike is nodding. But in fact, a lot of incredibly important work has been done in the last, 5 to 6 years determining high-resolution structures of the ribosome. And in fact, for those of you that pay attention to such things, the Nobel Prize in Chemistry just this last year was given for the structure of the ribosome. And some of the very first structure of the bacterial ribosome, the intact bacterial ribosome, was actually done right here at UC Berkeley by Jamie Cate and his group in MCB. And so I took an image from the work of Kate's lab and put it here in place of that little blobby cartoon, because in fact we know a lot at a molecular level about the structure of the ribosome. And I don't want you to get the impression that we just think it's a blob. We actually know in atomic detail what it actually looks like, right? So here's the large subunit over here. Here's the small subunit over here. Transfer RNAs come in the middle. You can see these little L-shaped molecules. And translation, protein synthesis is going on in here. And then the protein, when it gets made, it comes out the bottom. There's a pore, and the new polypeptide comes out the bottom. And we'll talk more about that later, but just to know that there's a lot of beautiful work that's known and a lot of structural detail about the ribosome. Okay, so the endomembrane system is the system that controls protein trafficking and performs a lot of the metabolic functions of the cell. And so this has a number of components to it. The nuclear envelope is part of it. The endoplasmic reticulum is part of it. The Golgi, and then various compartments that are called lysosomes, vacuoles, and the plasma membrane, right? So these are all membranous components of the cell. And very importantly, these are components that are either continuous, they're contiguous with each other, or they're connected by transfer vesicles. So these are little blebs of membrane enclosed particles that will pinch off, say, from the endoplasmic reticulum and then fuse with the Golgi. Okay, and we'll see there's some nice pictures of this in the book. And so what does this do? Well, it's very important for the biosynthesis of a lot of the molecules that the cell needs to use, and it also is important for getting those molecules to the right place in the cell. So again, the cell isn't just a bag of molecules. In fact, it's incredibly organized about where those molecules are made and then how they're utilized. So the ER accounts for more than half of the total membrane in a lot of eukaryotic cells. And again, remember, this is only in eukaryotic cells now, right? Not in prokaryotic cells. And it's contiguous with the nuclear envelope. How do we know that? Well, again, from micrographs, looking at these cells, and you can see when you slice through a cell that there are actual junctions between the ER membrane and the outer membrane of the nucleus. Okay, and there's some drawings that show this. And then there are two distinct regions of the ER, one of which is called the smooth ER, and it's called smooth because it doesn't have any bumps on it, and those bumps are ribosomes. So the smooth ER doesn't have any ribosomes on it, and in contrast, the rough ER are parts of the ER where we can see those little bumps, and those little bumps are the ribosomes. So rough ER means that there are ribosomes that are sticking on the surface. And here's some pictures again of this. So here, a beautiful micrograph here that shows the rough ER. You can see the ER membrane with these little studs along it that are the ribosomes that are stuck to the outside of the membrane. And then over here we see regions of the ER that don't have those ribosomes. So this is the smooth ER over here. And then again, here's a sort of an artist's drawing, and what you can see here is that here's the nuclear envelope, and you can see that the outer edge of that outer membrane of that nuclear envelope is actually connected physically to the ER. And that means that that it makes it very easy to traffic molecules into and out of the nucleus using the ER. So these are a few of the properties of the smooth and rough ER. So the smooth ER makes lipids. It metabolizes carbohydrates. If there are toxins that get into the cell, there are enzymes in the ER that will detoxify those molecules and try to get rid of them so they don't poison the cell. And it's also a place of site of storage of calcium. So calcium is a critical element in the cell, and there are storage sites in the ER for calcium. The rough ER has the ribosomes bound to it. These ribosomes are making and secreting proteins that are called glycoproteins. These are proteins that have sugar or carbohydrate molecules covalently bound to them. So they're called glycoproteins. And as we mentioned before, so the ER also pinches off these little vesicles. You can see one example right here that include proteins that are surrounded by this membrane. And this is a way of transporting those proteins to another site in the cell. It's also the membrane factory for the cell. It's where membrane is made. Okay, and so things move through the ER, and then eventually they get to the Golgi by means of these vesicles. So here's an example of a vesicle that's coming across, and the membrane is actually fusing with the membrane of the Golgi. And then the Golgi itself is a sort of a site for transporting and storing molecules in the cell. And so, when I was taking this equivalent course in college, in those days, the Golgi they were called Golgi bodies, and there wasn't not nearly as much known about the function of the Golgi at that time. And now we know that, in fact, the Golgi is, you can really spot it pretty easily in these micrographs. You can see it looks like this layer of membranes that are kind of pinched together, right? They're sort of like a sandwich that has many, many layers to it. And it's not just sort of thrown into the cell. It actually has a sidedness to it. So the cis face, which is over here, is the receiving side of the Golgi. And this is where incoming vesicles will fuse with this membrane and allow the contents to enter the Golgi. And then the contents will then move through the Golgi in a directional way, in the direction of the arrow. And then what's called the transface of the Golgi is the site where, again, vesicles are pinching off, and this is where molecules get incorporated into these vesicles, and they can then be pinched off and shipped to another part of the cell. Okay, a couple more types of aspects of this endomembrane system. So lysosomes, these are membrane-enclosed compartments that are important for digesting molecules in the cell. And we can see here's again a micrograph showing the picture of a vesicle containing two damaged organelles. So here's the vesicle that's bound by the membrane, and inside is a piece of a mitochondrion. And we can tell that because, again, you see these very characteristic, what are called cisternae. These are the structures inside of the membranous structures inside of the mitochondrion. We'll talk about that in a moment, but that's how we know that's a piece of a mitochondrion. And then here's a piece of another type of particle called a peroxisome. And these have both been enclosed in the lysosome, and they will eventually be digested, because the job of the lysosome is to contain enzymes that will break down these components into their more elementary units, such as lipids or amino acids, if there are proteins in there, nucleotides, and those can then be recycled and used to make other molecules in the cell. And a couple things to note here. So some types of cells will basically eat by engulfing other cells, and this is a process called phagocytosis. And when that happens, the cell will engulf the other cell and form and then the membrane will sort of pinch off and form what's called a vacuole inside the cell. And that material that's enclosed in there, the cell that's been eaten, will then be digested inside of that vacuole. So that's a process called phagocytosis. That goes on with organisms like amoebas. Maybe some of you remember, sort of a high school type biology class where you can actually watch amoebas eating this way. It actually is very important in human biology as well. For example, in the immune system, there are cells called macrophages whose job is to help kill off invading pathogens like bacteria by the same kind of process, phagocytosis, where those cells are engulfed and then digested by macrophages. The lysosome will come along and fuse with these vacuoles and dump its contents of digestive enzymes in there to digest them. The interior of the lysosome tends to be really acidic, and so those enzymes really only function very well in an acidic environment. If the lysosome breaks open and spills the digestive enzymes into the cell, they become sort of inactivated because the pH of the interior of the cell is much closer to neutral. Okay, so that's one way that the cell is able to control the behavior of molecules, by compartmentalizing them and then keeping the environment unique to the function of those particular enzymes. And one other thing I want to mention, because it's gotten a lot of attention in biology lately, is a process called autophagy. There's the word right there, autophagy, and this is it. Which is where lysosomes will recycle the cell's own organelles and macromolecules in a very orderly way. And so there's been a lot of interest in human biology and how this works, how it can be triggered, how cells can basically consume themselves or consume parts of themselves, and how we can control that process in terms of thinking about therapeutics for diseases like cancer, where we'd like to be able to kill off certain types of cells. Okay, and so finally I want to turn to organelles. So mitochondria and chloroplasts are the organelles in animal and plant cells that are important for changing energy from one form to another. They basically will take food in the form of either molecules that come in, or if you're a plant, in the form of light energy that comes in, and convert them into ATP so that can be used to drive various reactions in the cell. So chloroplasts are the sites where photosynthesis occurs in plant cells. Mitochondria are the sites where ATP is occurring in animal cells. ATP synthesis is occurring. And then peroxisomes, a third type organelle. These are organelles that are called oxidative organelles. They're sites where hydrogen peroxide is made and is used to break down other molecules in an oxidative process. All right, so a few things. Mitochondria and chloroplasts are actually not part of the endomembrane system, and you might think about why that is. Okay, think about what we said about the endomembrane system. It's all connected, right? These guys are separate. They're really separate units. They have a double membrane that surrounds the outer edge of their surface. They each have proteins that are made by free ribosomes. So there are ribosomes inside the mitochondria and inside the chloroplasts that are making proteins inside of those organelles. And they also contain their own DNA. Okay, and so because for those reasons, some of these properties, one of the really fascinating ideas in biology that hasn't really been proven, but I think most people are pretty confident that this is right, is that the origin of chloroplasts and mitochondria originally was from bacteria that invaded eukaryotic cells a long, long time ago and effectively became symbionts with those cells and now are really integral parts of those cells, even though they have their own DNA and their own ribosomes, right? Really cool. Okay, and then a couple more things about mitochondria. So these are found in virtually all eukaryotic cells. They have a smooth outer membrane, and then they have an inner membrane that's folded into what are called cristae. And remember, we saw those in that one micrograph, right? So very clear, when you see a micrograph, you can really tell what a mitochondrion because you can see those folded cristae stacked up inside the mitochondrion. The inner membrane creates two compartments, an intermembrane space and what's called the mitochondrial matrix. And then there's a lot of metabolism that goes on in there. Some of the steps of cellular respiration are catalyzed in the matrix. And then these cristae, again, are very important for creating a lot of surface area in the cells that are sites where enzymes are busily synthesizing ATP. And this is a picture. So isn't that gorgeous? Look at that. It's really beautiful. These cristae coming across, very clear. This is a mitochondrion that we're looking at. And there's a lot of nice surface area there for enzyme action to occur. And then here's the artist's rendition. Okay, and then chloroplasts. So these are a family of plant organelles that are called plastids. They're found in plants. They contain the green pigment chlorophyll and other enzymes that are important in photosynthesis. And they're found in leaves and other green organs of the plant, and they're also found in algae. Okay, so we can tell, very easily see them by their green pigment. And then finally, peroxisomes. So as we said, these are important for oxidation. So these are specialized compartments that have a single membrane on the outer edge. They make hydrogen peroxide, and they convert it into water. And the hydrogen peroxide is then, and the oxygen radicals that are made are used to break down different types of molecules. So it's a very important metabolic site in the cell. And finally, this is a great picture that just shows a combination of all three. So here's a chloroplast over here, a peroxisome here, and then a mitochondrion on one side here. We'll continue talking about cells on Wednesday.